All right. Welcome, everybody, to Standing for Truth. My name is Donny Budinsky. I am your host for this evening's program on why evolution is false. It is a privilege, as always, to have Dr. Jerry Bergman here with me for this important show. This is going to be comprehensive. We want to be thorough as we demolish the three pillars of evolution and work to falsify common descent. Dr. Bergman is a frequent guest here on Standing for Truth. His work has been a tremendous blessing to this ministry. I do want to encourage those in the live chat and those watching this program after our live show here tonight to check the description box for links to Dr. Bergman's articles, his books, and also his previous programs that he's done here on Standing for Truth. And again, we are going to be demolishing the three pillars of evolution. And we want this to be comprehensive, a go-to video for anybody interested or invested in the creation versus evolution debate controversy, as I have a lot for us to get through. We are going to be debunking, engaging, and addressing your major arguments for common descent and also your minor points as well. So if you're looking for that go-to video where you can get more or less all things debunking evolution, this will be it. And so before I hand it to Jerry, I am going to give my guest here a formal introduction for anybody who might be unfamiliar with him. Dr. Jerry Bergman has taught biology, genetics, chemistry, biochemistry, anthropology, geology, microbiology for over 40 years at several colleges and universities, including Bowling Green State University, Medical College of Ohio, where he was a research associate in experimental pathology, and the University of Toledo. He is a graduate of the Medical College of Ohio, Wayne State University in Detroit, the University of Toledo, and Bowling Green State University. He has over 1,300 publications in 12 languages and 40 books and monographs. His books and textbooks that include chapters that he authored are in over 1,800 college libraries in 27 countries. So far, over 80,000 copies of the 60 books and monographs that he has authored or co-authored are in print. I've got several of his books, but his book that we'll really be focusing on tonight is here. I've got it right in front of me, so people check this out. The Three Pillars of Evolution Demolished. It's comprehensive. It's a must read and also a must own. You're definitely going to want to add that to your library of creation versus evolution books. And so, Dr. Bergman, as always, it's great to have you for these programs. I really appreciate your time. I know how busy you are. And so tell us, how have you been and how was your Christmas, brother?
Well, I've been pretty busy as usual. I just got a paper done today. I find there's a website kind of like Wikipedia, except it's Rationalpedia. And they like to attack people like me. And so they did, and I responded to it today. Spent all day, probably shouldn't have spent that much time, but it's interesting the things that how they deal with us. One of the main things they do is name call. Yeah. And so if you have a case, the old saying is, you have a case, you present the case, present the facts. If you don't have any facts, you don't have a case, you name call. And so a lot of it's pretty obviously name calling. I'm surprised they can't see this, but they evidently can't. So anyways, today.
Yeah, no, that's great. Jerry, it's important, and I point this out, it's important we address the critics, we address our skeptic friends, and so I'm glad to hear that you've responded to that. I am familiar with that site, so I'm looking forward to that, Jerry. When that's ready and published, make sure to send that to me, and we'll share it around for sure. And so, I mean, as you said, name calling, that's usually the tactic or approach of those that have lost the debate, brother. And so I do want to give you a shout out, as many people are excited for this debate coming up in January. So we'll be having and hosting a big debate event here between Dr. Jerry Bergman and Dr. Dan Stern-Cardinal, both well known in the creation evolution debate world and community. And so this will be common descent or common design. Got to say, I'm excited for this one. And yes, I'll be moderating that debate. Jerry, you've got quite a bit of experience in debate, from my understanding. You debated P.Z. Myers in the past. Is that right?
Yes, I have. Yeah.
And how did that debate go?
A lot of name calling.
Oh, really? Eh?
Learned from the debates that you really have to just present your case and not try to refute his case. He said something like A, and I say, well, A is not true. The audience, they don't know what to accept. They don't know what's true or not. So I've learned you got to present your case and pretty much ignore what they have to say.
Well, you got to leave it up to the audience, right? It's up to the audience. Hopefully they're the ones that are analyzing and examining the debate, the arguments, the points being made in an objective manner. And so, Jerry, I am excited for your upcoming debate and hopefully a couple in 2025. Okay, well, what we're going to do right now is set the foundation for what we are going to be discuss. I'll give everybody a teaser. We are going to be engaging your most common and some of your lesser known arguments for evolution. That includes homology, nested hierarchies. We're going to address so-called transitional fossils, junk DNA, pseudogenes, ERVs, gene duplication. Those are just a few that we are going to engage tonight and give you guys some sufficient answers on those issues. But we're going to set the foundation and hand it over to Dr. Bergman to go over some slides and kick us off here in tonight's falsification of common descent. Jerry, the floor is yours. Go ahead.
Okay, thank you. First of all, some introduction, and that is research proves evolution true, but it's going the wrong way. So actually what's happening is de-evolution. And I need to define evolution, which I have, from molecules to man. Evolutionists often look at minor changes like the beak finches, things like that. And that's not the concern. Of course, all animals, or most animals at least, have a fairly large amount of genetic variety, genetic flexibility. So we're not concerned about small micro changes in the beaks of finches. We're concerned is from molecules to man, what you might call macroevolution, or basically evolution which produces all life. And the problem is not survival of the fittest. I don't really debate that because that's pretty obvious. If an animal is more fit, yeah, they're probably more likely to survive. The problem has always been, and this is a central problem with evolution, is the arrival of the fittest. Where does the fittest come from that natural selection can do its work? And that's what I focus on in this presentation. Okay, the primary basis of macroevolution, and again, I don't like that term, but you know what I mean now, is mutations. So to produce variation, there has to be a change in the protein, the DNA, and the regulation sequences. So these changes are all, according to evolution, a result of mutations. That's what they argue. And they admit that many mutations are harmful, but on the other hand, they say there's enough positive ones that therefore that can explain the source of genetic variety. Ernst Mayr, well-known evolutionist, he lived to be 99. Ultimately, he said all variation is due to mutation. Theodosius Dobzhansky, same thing. Mutation is the only source of the raw materials and hence of evolution. My throat tends to bother me. Okay, in the 20s, Hermann Muller discovered X-rays increased the mutation rate by as much as 100 times. So he figured, now we have proven Darwin. Now we know the source of genetic variety, and now we can actually speed up evolution by creating mutations by use of X-rays. And he did that. And basically, as a result, well, this article says it all, new discovery speeds up evolution. And this is in a science magazine. And basically, this is published around 1926. Basically, he says that We now know evolution works. We know how it works, basically by mutations. And he got a Nobel Prize for this, by the way. Not only is the accumulation, these are his words, of rare, mainly tiny changes the chief means of artificial, animal, and plant improvement, but it is a natural evolution, this is his words, has occurred guided by natural selection. Thus, the Darwinian theory becomes implemented, and this was from his 1946 Nobel Prize. This was part of his presentation he gave for his Nobel Prize. Richard Dawkins, professor at Oxford, and he explains evolution, and actually he does a good job explaining evolution. So we start out with a bunch of letters, just randomly typed in letters, and then and here's the problem, we program the system to select certain letters, namely, me thinks it is like a weasel. So we start with a W, well, that's not selected. The program selects M, so we get a M, and there it is. The next letter it selects is E, we get an E, and there we go. The next a T, and there we get a T somewhere here, there we go. And so with 44 changes, we end up with the goal, which is me thinks it is like a weasel. The problem is this is a complex computer simulation designed by very intelligent computer gurus. The problem with this is many. Number one, this is a huge problem. Mutations in the genome are almost always in what they call hotspots. And this is E. coli gene. Actually, the presentation I usually use uses P53, which is another important gene. And you can see that like 90% or close to that mutations are at this spot right here. And that is the 199 gene in the entire gene. So you count the genes and number 199, there you go. And that's mutated. And you get a few other mutations, and you get some that are very uncommon, and these are called cold spots. And this is very important in medicine because disease is caused, cancer, which is what I worked with, cancer is caused by mutations, and what you get is the same ones over and over and over. And this is important because we treat cancer according to the specific mutations that it causes. This is called tailored or focused cancer treatment. And we find that, again, with P53, it's a hotspot is a location. And that's important because when P53 is damaged, doesn't work anymore, what we have is about 10 times increased rate of cancer. In fact, of all cancers, about 50% are caused by damage to the P53 gene. And so we know huge amount about this, and we know why specifically mutations tend to occur in certain areas. Now, you'll never get selection when you have mutations occurring over and over in the same few areas. You'll never get Dawkins' goal, which is many, many changes. And of course, in his case, every letter changed eventually until he got all the ones he needed. But there we go. And of course, there are many examples of this. Another study, 2 mutations accounted for 94% of all mutations in the 319 they identified. And so therefore, hotspots will keep changing, but the other parts of the gene will remain the same. And another problem is the DNA mutation probabilities. Well, there are 6 different ways of getting serine. So therefore, if they were all random, looking at assuming they're all random, we would end up with 9.4% of serines changing from something to serine, or in this case, all these combinations produce serine. Arginine, 9.4%, leucine, 9.4, 9.4, and so on. Now we get to tryptophan. There's only one way of getting tryptophan. So what's going to happen? What's going to happen is if these were truly random, we would get more and more serines and fewer tryptophans. So if we take a functional gene, what's going to happen with mutations is it's going to deteriorate because we are going to get more and more serines more and more arginines, and fewer aspartic acids, glutamic acid, phenylalanine, and tryptophan. And so therefore, again, what we have is a mutation tendency to go downhill. It doesn't produce more variety. It simply causes deterioration of the genome. And we know this, of course, in cancer research. And so this is something that we look at. And then also charge and polarity families are important. That means for a gene to work, it has to have certain charge and polarity within the genome, because that's how they fold. That's how proteins fold and produce the proper shape, proper conformation. Well, we find that nonpolar uncharged, certain percentages are common, polar uncharged, certain percentages, but we're not going to get a gene unless we have the right number of nonpolar uncharged, polar uncharged, borderline, polar positively charged, and polar negatively charged. So we have to have this set. So what's going to happen in these changes? We're not going to get what we need, except rarely, very rarely, almost zero probability. What we're going to get is more and more of certain changes, more and more. So the result is more and more, say, nonpolar uncharged using a leucine. Okay, moving on. So when we apply this to our friend's set of data, start out with this, okay? And what we have is changes, and we end up with 40 billion changes. And in this case, we all end up with a bunch of T's. So it's not going to get ever what we're looking for. It's going to get deterioration. Now, of course, in life, it's not going to get up to 40 trillion, I guess, in this case. Result is hotspot and back mutations. Applying this to Dawkins' illustration, we're going to get, again, lots of changes in certain areas. These are hotspots. And back mutations, changes in certain areas. We will never end up with what we're looking for. And so, given the facts of genetics, we'll never end up with methinks it is like a weasel in this illustration of Richard Dawkins. Okay, are beneficial mutations common? Done a lot of research on that because I have an interest, of course, in finding if there are some, and there are some. The beneficial mutations often are a result of damage to one gene, allowing other gene to express itself. A good example is we have a few animals that are totally white. The fur is totally white. What happens is the genes for color are broken, and therefore the color of the fur is naturally, without these color genes, white. And so we have an interesting new trait, but it's not a result of mutations. It's a result of mutations damaging the control gene, allowing the other gene to be expressed. And I did literature search. Total mutations, I came up with about 500,000. And this is from abstracts of major abstracting services. And so 500,000, and of those, they claimed there was 186 beneficial mutations. So there were some. We're talking about 0.04 were beneficial. And let's look at some of these beneficial mutations. One of the main ones was seedless fruit. Well, that's not beneficial for the fruit. It's beneficial for humans. So many beneficial mutations were actually not beneficial for the animal or the plant in this case, but they were beneficial for humans. And a lot of these beneficial mutations are beneficial in this way. One of the most interesting ones is the Belgian blue cattle. This was discovered in Belgium. And what we found with this mutation was you get about 30% more muscle. That's great. The meat's tender and it's low in fat. So we're talking about, for humans, an ideal change, which is very valuable. Well, it turned out it wasn't very valuable in the long run, because what you end up with a cow, which is the Arnold Schwarzenegger of the cow world. And that basically is, the result is this is breaking the brakes, which stops muscle growth. Muscle growth has brakes, which allows it to develop only to a certain point, but this mutation breaks the brakes. Brakes don't work, so as a result, the development of the muscle is much, much greater. And this turned out to be so problematic for the farmer that even though for sales, for selling their meat, it was beneficial, in the long run, it turned out to be not only not beneficial. Reduced fertility was one problem. So it produces beneficial effects for the farmer, but because of information loss, it doesn't produce in the long run total beneficial result for the cattle. And they know now what it caused by homozygous deletion for 11 base pair coding regions. So it doesn't produce the brakes because the system that codes for the brakes is broken, doesn't work. And so many of these examples are not beneficial for the animal. And in many cases, not even for us. One of the most famous is a 1791 mutation Seth Wright noticed. And this was in animals that had short legs. They resembled dachshunds. And as a result, they couldn't jump low fences. Okay, and so therefore, it was an advantage to the farmer because he didn't have to build fences very high because they couldn't jump even somewhat low fences. He didn't have to build fences very high, and they would keep the sheep within a certain area. And again, we can see a picture of them. We can see why a fence about 6 inches high would stop the sheep. But here we can see a normal sheep. A normal sheep in between is the uncon sheep, which is about 4 inches off the ground. This is one of the most famous examples. Darwin talked about this, and this is one of the most famous examples used in textbooks for years. And this is actually a picture I got from a high school textbook. And so this is called a ancon breed. Okay, but in fact, what it is the sheep had a disease called achondroplasia. And it was a diseased sheep. And best example of that, of course, is in people. There's a young man with achondroplasia, and this person happens to be an actor. And as a result, for certain roles, he's in demand. And how do they deal with this? They deal with this because, some people can get jobs as actors who have this problem, but most of us don't want our kids to have achondroplasia. So how they deal with this is they break the legs, they pull them out, they heal, they break them again, they pull them out by braces, and then they heal again. And so they can end up with someone not quite as short as this gentleman here. And I presented a presentation at Ohio State University, and the professor who was there said, "You must be wrong. There must be some beneficial mutations for humans." And I said, "Well, yeah, I'd like to see some because I'm sure there are." He looked and he looked and he looked. Finally, he sent me this picture. This is a man who has 6 fingers. And he said, "This is beneficial because this guy can really climb up trees very, very effectively." So there, he said, we have now a case of one beneficial mutation. Well, actually, I knew about this because we have a lot of Amish people here, and that is a common problem among the Amish. Unfortunately, though, it's not a nice, neat 6 fingers on any hand, as we can see here. Unfortunately, and this is one case, unfortunately, it's more like this. Generally, what they have to do is snip off the extra digits because they're not helpful. They're in the way. You cannot behave normally in a normal environment with this digitation mutation. And this is another example you can see. And this person is going to be handicapped because of this mutation. And here this child has an extra arm. I mean, that would be very useful. Think of mothers who are trying to take care of their kids and cook supper. This would be really handy to have 3 arms. Unfortunately, though, in this case, the damage was such where the one arm was non-functional, and I forget which one, but one of these was non-functional, and they had to lop it off. And so therefore they had a problem with it. Sheep with 8, 9, 10 legs. Yeah, that brought in money for the farmer. It was a oddity that people came in and paid 25 or 30 cents to see. But on the other hand, this sheep could not walk. And so without being taken care of by the farmer, this sheep would perish in a environmental situation. And again, there are lots of examples of mutations, but again, I'm not saying there aren't none, but I have yet to find one really good example of a clearly beneficial mutation for humans. And these are a couple of cases of double heads. And this is a case where a child was born and didn't separate during a certain period of development. This case right here is where a man has a disease which he produces a very HER2 condition. And in some environments, I guess this might be beneficial. If he lived in a very cold environment, it might be helpful. But by and large, in most of the environments that I'm and you're familiar with, that's not going to be beneficial. And here, this child has a huge head. It's not a genius, unfortunately. The abnormal development caused the death of this child. And here's another example. There are lots of examples of mutations. And when I mentioned at the medical school about favorable or beneficial mutations, the only response I got was the look. Are you familiar with the look? Kind of like, what's wrong with this guy? Not a word was said, but just the look, because I was sincere. I wanted to know, is anybody in the department at the medical school, was anybody there aware of a beneficial mutation? Well, didn't find any. And there are just lots and lots of abnormalities caused by mutations. And here's one beneficial mutation, which is seemingly beneficial for a certain area, and that is streptomycin. Fits in here, and the ribosome there doesn't work. Okay? When it fits in, it's the control, it's a regulation system. And streptomycin here actually turns off the ribosome, so this is an antibiotic for people who have certain infections that streptomycin cures. Now, the bacteria can get, as a result of a mutation, it gets distortion of the receptor, and therefore it doesn't work. And so this bacteria now is resistant, and they say it evolved. It didn't evolve. Its damage resulted from this change, and as a result, it's now immune to streptomycin. But as you know, this damage is important because this regulation is critical for the bacteria. Once this is damaged, the regulation is lost. And yeah, it's resistant to streptomycin, but in the long run, it is not an advantage to the bacteria. And there are many other examples of this. And again, these are caused by mutations, but they're not in the long run beneficial. Here, this is a T cell. And for the AIDS virus to get in, it's got to have two keys, one here, CD4, and one here, the CCR5. So it cannot get in the cell and cause AIDS unless it has both receptors. And this is the T cell, which has both normally. But when this is damaged, in this case, CCR5, is damaged, it isn't full. The whole thing doesn't produce. It's not all sequenced. And as a result, it's a partial, non-functional CCR5. So as a result, HIV in this case cannot bind. And because it cannot bind, guess what happens? The person with this mutation is immune to HIV. He can be exposed to it over and over, and he is not going to be affected by it. But as you may expect, this damage may produce an advantage in resistance to HIV, but in the long run, it is a problem because the person has a less functional immune system and is more prone to certain diseases like liver cancer. And so, yeah, it's beneficial in a very unique way, but in the long run, it is not beneficial for the person. And so Shermer estimates, he's an evolutionist, trillions of distinct modifications are required to evolve humans alone. Yeah, that's fine. But on the other hand, if 99.9% are damaging, you will never end up with evolving humans. And actually, the vast majority, about 99.9% of all mutations are repaired. So even if you got a beneficial mutation, it would be repaired and it wouldn't show up in the offspring. And so therefore, there's a problem. The only ones that show up are those that are not repaired or actually not repaired directly. And those are the ones that cause problems. And therefore, evolution is not going to work, clearly. To me, it's not rational. Now, how do evolutionists respond to this? Well, they respond primarily by evolution is true. It has to be true. Yes, Bergman, you have a problem. and we cannot solve it, but we know evolution is true. Therefore, there must be some solution. My response is that you can't have evolution until you have a means of producing genetic variety. And Darwin himself had no clue, and he realized that was a problem. How do you get genetic variety? He didn't know. And today we still don't know. And as a result, if you don't have genetic variety, you don't have evolution. So evolution is stopped from the get-go. It cannot occur. You've got to be able to get genetic variety. And if it cannot occur, evolution cannot occur. So all the so-called proofs of evolution can be explained in some way, but they can't be true as causing evolution until we get a source of genetic variety. And I can mention there are some mutations that are near neutral. That means they don't cause problems by themselves, but the damage adds up. It's like a car. The car has leaks oil, but it still runs. The spark plugs are dirty. They need to be tuned. It still runs though. And after a while, you get so much damage. After a while, you end up with a car not running. The same is true with people. As we get older, the deleterious mutations accumulate, and that's called aging. And as a result, people, when they die, in most cases, especially when they're older, they don't die of anything that causes the problem. There's not one thing. It reminds me when my aunt died, I asked the doctor what happened. She was perfectly healthy. As far as I know, the only time she's been in hospital in her life was when she had her son. So I asked the doctor, what happened? What did she die of? And he said, well, no, she didn't die of anything, really. Wait a minute. She's dead. She died of something. What did she die of? And he said, well, here, Bergman, sit down. Let me explain to you. She died of actually nothing, but her lungs weren't working quite as well. Her liver wasn't working quite as well. Her nerves weren't working quite as well. She had a lot of near neutral mutations, which added up. And when you add them up, you eventually always result in death. And so he could say she didn't die of anything, which is true. Or he could say, which is probably more accurate, she died of everything. So what did he put on the death certificate? I noticed he said myocardial infarction. I said, why? He said, because we know in the end, when you have all these problems, the heart gives out. So therefore, we don't want to fill out the whole page and all these things if she had problems with her. So we just write myocardial infarction, end of story. And people understand that. They understand that she didn't die of any one thing in many cases. Now, there could be she died of one specific thing, but in most cases, when they die of old age, what they mean is they died of everything, and therefore that's why she died. She was, I think, late 80s, early 90s. But at any rate, I go into more detail, but that's the heart of this presentation. And any comments?
Questions? Yes, lots of comments, Dr. Bergman. As always, comprehensive, technical, which is necessary when we are engaging and debunking the best evidence for evolution, that kind of evolution that says all life, including plants and animals, are related. And so, since you helped us set the foundation there and touched largely on so-called beneficial mutations, you did an excellent job there, Jerry. Here's a few of the most common ones that you'll see, the evolutionist favorite. Now, one thing I'll say, just as a preface, I find it interesting because even if, Jerry, and feel free to tell me if you agree with this, even if they could show us 1, 2, even 10 truly beneficial mutations that are not functionally compromising or reductive in any way, that isn't going to counterbalance the damage that you have described based on the accumulating genetic load through nearly neutral mutations that are being passed on every single generation. They're slightly deleterious. Selection can't see them, and therefore they build up over time and result in genetic sickness within species. And so even if they could show us a truly beneficial mutation, wouldn't they need millions of those to counterbalance the damage being done, Jerry? Oh. Oh, Jerry, I think is that just on my end? I'm not getting any sound. Maybe oh, I can hear you now.
Can you hear me now?
Yes. You're good to go. Yeah, Jerry, I was okay. Looks like you were frozen there. I was wondering if it was my end, but the audience says, but it looks like you're back. So, Jerry, my question was essentially the evolutionists, proponents of common descent, even if they were to provide a truly beneficial mutation that wasn't reductive in any way, is that really going to help them in counterbalancing the damage being done through genetic degeneration?
These examples which you gave, for example, sickle cell anemia, what happens is the cell's damaged, and as a result, it's interesting that the problem is the malaria bacteria cannot affect it because the cell is partly damaged, and I think it's only the sickle cell trait that's the problem. It's not the sickle cell disease. That's because that can be lethal. In the long run, it often is, but without treatment. And so therefore it's a horrible disease to have. But the trait, you don't have the disease, it's not a problem typically, but you also have some immunity against the problem which causes the malaria. And that's an advantage, of course, if you live in Africa, where malaria is a common issue. It's not an advantage here much because we don't have much exposure to malaria. But yeah, you can damage things partly, and it can produce a benefit, but it always produces the other side, the problem which seems to come with a benefit. It's like when you have a car that's all rusted out, that is not a benefit when you're trying to pick up your date. But on the other hand, if the lending company wants to repossess your car, fine, take it. So it's an advantage in some situations, but it's not an advantage in other situations. And so that's a good example of these. But it's interesting that all these examples you showed over and over found. Mm-hmm. It's interesting that these are the only examples they can find. I mean, there must be millions of examples, or at least hundreds of good examples. But all the ones that they show over and over are problematic. And I see that as a big problem.
Right, because you can just do a little bit of research on the harmful effects of mutations, the deleterious nature of mutations, what diseases they cause, and you're going to find tens of thousands of examples.
Yeah.
And so I do find it very interesting. I find it curious that it's really the usual suspects. It's the usual 5 to 10 that we could debate all day, but there should be no question. There should be that many truly beneficial mutations out there, Jerry. So they'll say, well, you guys don't understand what a beneficial mutation is because that's exactly it. It's context dependent. In one environment, it's beneficial, like sickle cell anemia. It's beneficial to those who have malaria, but it's deleterious in another context. So they'll say that's what we mean by beneficial mutation. But is that really helping them in any way, Jerry?
Well, that's a wordplay, I think, and that doesn't help them. What you need is you need the ratio between beneficial and deleterious mutation to be a lot different. You know, even if there's only 1% of all mutations were beneficial, or even a quarter of 1% were beneficial, that would be a strong argument in their favor. But that's not what we find. It's not 1%. It's less than 1%. And it's 0.00001%. And so when we have, and as you brought out, your nursing background, so many diseases, about 5,000 diseases now are known to be caused by mutations. The main one, of course, is cancer, and you've got many different types of cancer. You have skin cancer, you have liver cancer, you have kidney cancer, and so on. And so cancer's a major killer. Number one or number two, heart disease and cancer vie for number one place, but still, and a major cause of heart disease. Other causes as well with heart disease, but a major cause of heart disease is mutations. It's an inherited defect, which you end up with more prone to certain problems, heart problems. So yeah, you're right. It's just, I guess when they're desperate, they're going to argue for their worldview, and it's a worldview. And there's a film that a friend of mine did, and he's a lawyer in fact, I got his name right here, John Calvert. He did a film. He had a meeting in Kansas about the whole idea of creation, evolution, intelligent design. And he had like 10 scientists present the case for intelligent design. And there was a lawyer there. And this lawyer was so incredibly nasty and mean and angry. He was really angry. I thought he was going to have a stroke. He was so angry. And you have to wonder why in the world these people were presenting their viewpoint. And in many cases, they weren't disputing evolution. They were disputing Darwinian evolution. Like Michael B., he accepts evolution. He's not an evolution denier, but there's a problem in what evolution can do. And even when you present that pretty soft evolution, he got so angry, and I just couldn't understand why this man was just so out of it. Well, this is his worldview. This is the way he sees the world. And we're basically disputing his worldview.
Yeah.
And that's why he got so angry. He did not want that worldview to be questioned.
Well, they've got their basic presuppositions according to that worldview. And so rather than tapping out, they need to come up with a rescue device to preserve that worldview, Jerry. And what you said on the nature of beneficial mutations and sickle cell anemia, to be exact, it brings to mind an illustration. It would be like you had two warring countries fighting each other, and one country is sending their tanks over the bridge to get to the other country that they're fighting. So the country, as a defensive strategy, decides to blow up their own bridge, and that way the opposing army's tanks can't make their way across. Okay, it's beneficial in that context, but they're destroying their own bridge. It's not explaining how the bridge was created or engineered. No, they're destroying the bridge for a short-term benefit.
That's true. That's what Germany did. They destroyed all their bridges but one. And that one, American Canadian troops just crossed very effectively.
Bombarded.
And
so that doesn't tell us how Germany built their bridges, does it? Tells us how they destroyed their bridges. So another big example, I'll put it up on screen again, and we'll make sure that there's no freezing this time. So let me know that it's coming in okay. You addressed the HIV resistance one in your presentation just now. A popular one that's brought up in many debates, more informed evolutionists, I guess I'll say, they like to use nylon digestion by bacteria. So this one, Jerry, if you can see it, they'll say, oh, well, these bacteria have gained a beneficial mutation or a series of beneficial mutations that have allowed them to digest synthetic nylon. And so that's a beneficial mutation. That's a true novelty, they'll say. What's a good response to that one?
Well, it is a mutation that changes things, but of course, enzymes, which is what we're looking at here, enzymes are often pretty flexible. Small changes can produce the ability to respond in very different ways than other enzymes. And that's basically what happened here. And it could have been a mutation, but nonetheless, it was a small change, and that allowed it to break down nylon, which normally couldn't do before this. But nylon is a carbon compound, and it's not all that different structurally than many compounds that they ingest. And so I'm not sure this is usually proposed, but it could well be that this enzyme has the ability to produce slight changes, and as a result can digest compounds it couldn't before this. Right? We have new carbon compounds, I talk here, we have new carbon compounds developed all the time. And fortunately, in many cases, our enzyme system can break these down. And that's good. And I think part of it's just the flexibility that we have in enzymes being more flexible, being able to break down similar compounds very effectively.
Yeah, I think those are some excellent points. In my research, I've discovered that there seems to be a bacteria from a diverse set of environments that have this pre-existing ability, Jerry, to digest nylon waste products. So it seems like that ability is already there, built in. It just needs the right environmental stimuli to manifest.
That's a good example, because one major function of bacteria is to recycle. And we know bacteria can recycle just about everything, given enough time. And therefore, that illustrates indeed that they have the innate ability to be able to break down anything new that we come up with. Nylon and anything else we come up with, eventually they can break it down. So I feel that was part of the design of bacteria in the first place.
Yeah. Yeah. You know, it's forward thinking. It's flabbergasting to me that these are the best examples put forth to have us believe that fish evolved into fishermen, particles evolved into people, goo to you, bacteria to biologists. These are the best they have. They're going to have to offer something a little more inventive than that, right, Jerry?
The key is, though, they really believe. They have a strong, there's a book, I forget the author, but it's a book, I Don't Have Enough Faith, that's the title. I don't have enough faith to be an evolutionist. And in many ways, to be an evolutionist takes a lot more faith than what we have, and they often call our worldview a result of faith. Well, it is, but evidence is very important in our worldview. And for evolution, in many ways, faith is far more important for their worldview than it is for our worldview. And these examples we've been talking about are good illustrations of that.
So here's a new one that's come about recently. It's come about in a debate recently, and they're always going back to the drawing board when their favorite ones are debunked to try and find one. Well, what about this one? What about that? Like, they're kind of just throwing everything at the fridge, seeing what sticks. So a new one, and this comes in from a commenter, Sydney Raptor, appreciate it. The relatively new beneficial mutation tetrachromacy is also a great example. So apparently it's rare, but there's a certain subset of people, I think strictly women, that have this mutation that allows them to see more colors because they have more cones, or they can see colors more prominently. I'm not sure if you're familiar with this one, Jerry, but if you had any thoughts on it.
Yeah, again, one thing that you notice when you study life is that there is really quite a large amount of flexibility. And this, I think, is one example of flexibility, which is obtained as a result, and it could be a mutation. It could be a gene is broken and therefore allowing this trait to develop. I'm not familiar with all the details with this example, but on the other hand, we're looking at so many small things. Now, of course, for the animal, this tetrachromacy may be important, but on the other hand, we're looking at the best examples, I think, is where birds come from. Birds evolved from dinosaurs. To go from a dinosaur to a bird means a lot of steps. And now they're looking for dinosaurs that have possible evidence of feathers. So they're very excited because, now we can see dinosaurs will eventually become birds. And another example is, of course, whales, where the whales came from. Well, whales evolved from a dog-like animal. So how do you get a dog-like animal to a whale? We're not talking about small changes in their perception of colors. We're talking about enormous changes to go from a dog creature to a whale. Or another example is, how do we get dogs in the first place? We go from fish to tetrapods, and therefore we're talking about major changes, not these minor changes that I think you find. So many of these minor changes, I think they're built into the design of animals. It's like dogs. We have now something like 500 different breeds of dogs. How do we get all these breeds of dogs? Well, my conclusion is that The dog gene system is built in to produce variety that we can get if we only breed long enough and are breeding correctly.
Yeah, it's due to the pre-existing ability to change. The second God created organisms to change, that means the so-called evidence that Darwin presented in his book on the origin of species are actually points or lines of evidence for the creation model, because it's just basic adaptation, and change over time makes sense. But these are horizontal changes, aren't they, Jerry? They're not vertical changes like the evolutionists would need.
That's true. It's like they're doing surveys of melanin, and it's surveys are indicating, now this is partly due to intermarriage, but surveys are indicating that more and more blacks are, as they live in America, becoming lighter. And it could well be, that's what they indicate, could well be that in another 300 or 400 years, most people who are, quote, from black families will become maybe brown, light brown, or even lighter than light brown. And when you compare, and they do this by comparing American blacks who've been here for 300 years to blacks in Africa. And you notice there are definite color differences between blacks that came over recently from Africa and blacks that have been in America for so many years. Now, part of that's intermarriage. That's true. But on the other hand, the perception is that in time, the skin color will change due to the needs of the environment. And in America, we don't have the same needs to be protected from the bright sun that you did in Africa.
Interesting. So if I'm understanding you correctly, does environment dictate a lot of the phenotype, the adaptive episodes that could take place in human species? A lot of it's environmental stimuli?
I think, yeah, it is. A lot of it's environmental stimuli, and that's why we see so many differences based on where people live. Yeah. People who live in very hot areas tend to be short and fatter. Sorry, people who live in hot areas tend to be thin and thinner because they can dissipate heat better. People who live in very cold areas, such as the poles and Greenland and so on, tend to be shorter and fatter. And I would conclude from this that indeed, as people lived in hot or cold areas, after hundreds and hundreds of years, they end up as a whole changing slightly. And that's why in certain parts of the country, the hot parts, we have tall, thin people, and in cold parts, we have very short, fat people, because it does protect us from the weather. And I would say that's one more example that we do have some adaptability. But the people who are tall and thin, again, there's limits as to how much change can be produced from this technique.
Right. It's not unlimited variation, but there is a lot of morphological adaptability built into each original created kind in their groups. And so historically then, people groups who lived closer to the equator, if environment is dictating a lot of these changes, could those people have accumulated more mutations because of the negative effects of the environment they live in historically, Jerry?
Or I don't know if it's a result of mutation. I think it's a result of built-in adaptability.
Okay.
The built-in adaptability in time may represent itself slightly in 1 or 2 generations, but after 8 or 9 generations, it expresses itself much more. And notice the same thing with dogs, and how dogs who've been pets for decades are in many ways changing their personality. That wild dogs who have not been adapted, who have not been raised as pets, tend to have a different personality than dogs that have been pets for many, many years. And I think that's a result of the environment.
Yeah.
And the ability to adapt to the environment.
Those are some good, interesting points. And with the tetrachromacy, I did a little bit of research on this because I'm always curious when proponents of common descent, guardians of evolution, bring up the big new example. I always think to myself, okay, what negative effects does this so-called mutation have? And so I did a little bit of research, and apparently Firstly, as I said earlier, it's rare, but people that have this tetrachromat, they're actually more sensitive to light. They're more sensitive to the sun, computer screens. So there seems to be a downside, as usual, something negative associated with these mutations. Even though there might be a benefit, like, oh, I can see colors more prominently. Unfortunately, now they're more sensitive to certain colors, like yellow, from my understanding, and also more light sensitive. They see a lot of starbursts. So again, as usual, right, Jerry, there's negative effects associated with these.
They may fit well into a paint factory, analyzing the color differences. So in certain environments, it may clearly be beneficial.
Yeah, these aren't going to take your particles to people, nor are they going to counterbalance the damage done by the accumulating genetic load. So here is a student of evolution in the chat, and we do appreciate our critics and skeptics. And this is more or less one of the main arguments or rebuttals to genetic degeneration, Jerry. So he says, near neutral mutations. So again, nearly neutral mutations being those low impact mutations. Jerry, you pointed out it's kind of like rust on a car. On their own, they're inconsequential, but they build up over time and the car degenerates. It gets destroyed. You eventually get enough rust where the car is not workable. Or I like the illustration that it's akin to a single spelling mistake in a book the size of an encyclopedia. On their own, they're inconsequential, but enough of them build up over time and the book is now gibberish, hard to read. So these nearly neutral mutations, not subject to selection, they build up. Their counter is this: they can only build up due to drift. Well, yeah, if selection can't see them, they're then subject to drift. So they drift and they spread that way. He says it's very slow in larger populations. They're quickly overwhelmed by any rare beneficial mutations that appear and reach fixation. You see that this is what they try to do, Jerry. They try to say, well, beneficial mutations are going to counterbalance the damage. My quick thoughts, and then we'll get your thoughts. Beneficial mutations are rare. They're about 1 in a million. Lenski's experiment demonstrated that. But when they do happen, they're usually reductive. As you've been pointing out, they're functionally compromising to organisms. So there's no real trade-off. If anything, what you're going to get is what Lenski, I think, demonstrated in his experiment. You're going to get shrinking functional genome sizes. You might get a linear increase in fitness, but in total functionality, it's a decrease. So I think this is more of an assertion, and they need to prove that assertion, Jerry. Anyways, what are your thoughts on this counter to what we're saying?
Well, drift is talked about a lot, and what they seem to do is that they extrapolate from what we can see to, well, after a million years, we get slow drift, we end up with people, or we end up with, from dinosaurs, we end up with birds. Well, that is problematic because we're talking about so many changes. And the problem is these changes have to be, in essence, small enough so that the animal doesn't become extinct. As a dinosaurs evolved into a bird, it's got to be able to survive and compete with other animals during this time of change. And therefore, it's a way of explaining things, but it really doesn't prove anything because we still have to produce the changes that are necessary without producing problems. And of course, drift can produce problems. And so therefore, how do we know it's going to work in the direction toward, in the case of dinosaurs, in the direction towards birds? And of course, evolutionists are talking about belief. They believe it's true, and therefore they look at the world with their evolutionary glasses and they see what they want to see to some degree with those glasses. Whereas if they would take the glasses off, they would see things in a very different way. In many ways, creationists don't have the glasses on. They take them off. And therefore, they see the world for what it is. Where evolutionists have to see the world relative to change. And therefore, they have to have the evolutionary glasses to see what they want to see.
Right. So if they have their evolutionary glasses on, they can't see the reality that these deleterious mutations They go unchecked. Selection can't see them. And therefore, they put shelf lives on genomes. That fits beautifully with the biblical creation model that Adam and Eve were created with no mutations. They were created with design diversity or created heterozygosity. Deleterious mutations are introduced after the fall. They're introduced to quite a large extent after the flood. And so we've been going downhill and degenerating from a once perfect creation. That fits. That's what we would expect. But because they have their evolutionist glasses on, as you're saying, Jerry, they have to actually look at this and say, well, no, maybe this could solve the problem, or maybe this could solve the problem, beneficial mutations, right? Because they got that worldview that they need to preserve, right, Jerry?
You're right. And we talk about mutations. Actually, of course, somatic mutations are not what we're looking at. We're looking at germline mutations. And that's an area that's in many ways whole different than what we know about somatic mutations. In germline mutations, it doesn't take very many, and the sperm or the egg can't fertilize properly, can't produce a zygote. And so therefore, it's in many ways the problems that we see with somatic mutations are really much worse with genetic mutations found in the gametes.
It's funny because genetic drift actually makes the problem worse for them. I wrote a technical article with a friend on this, and we had it reviewed by Dr. John Sanford before publishing. And in the literature, you'll find from people like Douglas Fatuma, they'll point out that your neutral mutation rate, Jerry, equals the long-term substitution rate or fixation rate. So if you have nearly neutral mutations that are slightly deleterious reaching fixation according to the evolutionary model, that is irreversible damage that's now stuck in the entire species, in the entire pot, because that substitution is species-wide change. So sure, they can have these nearly neutral mutations reach fixation. That's just permanent damage in the population. Makes the problem worse for them.
Part of the problem they have and I had was it took a long time, in my own experience, to crack the egg. I knew about this mutation business 45 years ago, and it didn't gel. I didn't comprehend the implications of it. And I could say 40 years ago, even when I was an evolutionist, yeah, most mutations are harmful and it's problematic and it's very few beneficial mutations, et cetera, but it really doesn't gel until you think about it, until you reason about it for some length of time, because in many ways you're changing the worldview. And I had to change my worldview, and that took a long time. My guess is most evolutionists, if they want to change the worldview even, it's going to take time. That you can't accept it right away. And you have to keep, in my case, I kept reading and reading and reading about mutations and evolution and areas such as that, and it took a long while before it all gelled together. And once it gels together, you see there's no way evolution can occur. It cannot occur. And even though I knew the right words 40 years ago, it didn't gel so that I accepted the fact that evolution's impossible. And that took a long time.
Right. Jerry, excellent points. So before we move on from beneficial mutations, I said at the beginning I want to leave no stone unturned, and so I want to stick to that. The other big one that you'll see, and they'll never tap out on this one, it seems, and you've addressed this so we did a show together, Jerry, about a year ago on mutations. We did 2 hours strictly on mutations. Excellent program. I recommend people check it out. So this one here, the long-term evolution experiment with Lenski's E. coli bacteria, they say that experiment demonstrated several examples of truly beneficial mutations. They'll say evolution in action. Is that really what they observed, Jerry?
No, creationists love that experiment because it supports our worldview very effectively. And so therefore, creationists talk about that a lot, because here you're talking about large number of mutations, long term. Of course, bacteria, we're talking about, they're here and today and gone tomorrow. So their life expectancy is, you want to call it that, very short. And so therefore that is a an argument in our favor because, yeah, you had a few changes, but by and large, you didn't have any significant evolution. You had simply the ability to consume different nutrients in the environment, which changed slightly. And I forget which one it was, but it changed the ability to deal with certain environmental conditions.
Right. It's also an answer to, and I agree with you, Creatious love Lenski's experiment, because an objection to genetic degeneration, Jerry, is typically, oh, well, if genetic entropy is true, then why are there still bacteria? Bacteria should be experiencing genetic degeneration. And yet that's exactly what we've discovered in Lenski's experiment. If I understand correctly, the genes in these E. coli bacteria over the many generations that they were looked at, they were deleted, broken, downregulated, or even just completely eliminated. And so the type of adaptive episodes that we see occurring in Lenski's bacteria would be akin to, if I wanted, Jerry, temporarily, if I wanted better gas mileage in my car, I could start removing weight from the car, basically destroying the car, remove the side mirrors, maybe remove a couple of the seats, the internal seats. Okay, so temporarily I got better gas mileage, but the car's shrinking, I'm destroying it to get that better gas mileage. And that's what we're seeing in Lenski's bacteria. They've now shrunk in functional genome size. They're lazy, they're handicapped. They're not really any better, are they?
Yeah, that's true. They're not.
And they're still bacteria.
I'm surprised that he did that experiment because he must have known that in the long run, it could be a problem for evolution, or at least perceived. I'm surprised his colleagues didn't say, no, wait a minute. This may not turn out like you think. This may end up hurting us more than helping us. And I think in the long run, that's what happened.
Right. He provided ammo for us. And they like to do that. Just like I was doing a little study last night on mitochondrial Eve and Y chromosome Adam. And it was actually the evolutionary community that first discovered our first parents, the first couple, the fact that we've all descended from two people, Eve and Adam. Of course, they don't want to, because of their worldviews, you've pointed out so brilliantly, Jerry, they don't want to concede and say, this validates the Bible. So they put a spin on it. But they're the ones that first coined those terms, because they're the first to say, whoa, the mitochondrial DNA and Y chromosome DNA data suggests we came from just two people. So they're discovering and helping us to validate the Bible, aren't they, brother?
Well, they would explain that simply due to a genetic bottleneck.
Right.
So we had lots of variety before that, but the bottleneck, there was only 2,000 or 3,000 left, and we all descended from that small bottleneck, and therefore it appears that we descended from two people, but we didn't, because the bottleneck explains what we find.
Right. Right. They want to say, well, okay, the low diversity in humans, we're all 99.99% the same. We must have come from a small ancestral population because of a bottleneck. Like you said, it's post hoc ad hoc, after the fact explanation. Because if you think about it, Jerry, take Homo erectus. You've done a lot of work on hominins. Take Homo erectus. I did a little research, and they believe Homo erectus was the most successful pre-human hominin that has ever existed. So they say erectus existed on the planet across Eurasia. They left they were the first to leave Africa, according to them. And they existed and flourished here for 1 to 2 million years, and may have existed in a population of about 1 million across Eurasia. And okay, my question is, where's all the dead bodies? Where's all the billions of dead bodies of erectus if they existed on the planet for 1 to 2 million years? Say they existed in a population of 1 million. Let's say their generation time was 20 to 25 years. Where's these billions of dead bodies? As far as I know, all the Homo erectus fossils that we find can basically be fit into the trunk of a car. And so it doesn't really look like their evolutionary story fits reality. Is that right?
Yeah, that's a problem that they are working on. But it's a good point, because they realize that given all these millions of years and huge populations, there must be enormous number of bones out there. And of course, they don't find them. Now, one explanation is, well, they don't preserve very often. So 99.99% were not preserved. And therefore, that's why we don't find them. But on the other hand, given the large number that must have existed, even if it's only 0.01% that are preserved, there still should be a lot of bodies out there. And the ones they do find are in a hole 3, 4, 5 feet deep, and they're preserved pretty well. So if some were preserved pretty well, why were not more of them preserved pretty well? And of course they weren't, and so they claim. So therefore, that's a problem.
Isn't that an argument from silence? Oh, well, fossilization's rare. Yeah, that's our whole point. That's why the majority of fossils we find is due to a worldwide catastrophe known as the flood. That's how we can get so many fossils in the first place. And then, so basically you have humans existing for all these years, hundreds of thousands of years, but not until 5,000 years ago, these humans, which even the evolutionary community would agree that Neanderthals were sophisticated people. You've given a presentation on that. Erectus were a sophisticated group. They admit they had the ability to sail, make fire. So only 5,000 years ago, they just figured out agriculture, figured out that putting a seed into the ground would be beneficial. And, you know, because all within 5,000 years, we see the history of mankind. We see recordings, historical documentations. The evolutionists, correct me if I'm wrong, Jerry, they have to say, well, for hundreds of thousands of years, humans or hominids simply did not think to invent agriculture.
Yeah, that's of course one problem I have with the timelines is that when they come up with problems in the evolutionary record or whatever, the solution is just add more years to the dates. So we see a problem here. So instead of man evolving 6,000 years ago, it's 10,000, or instead of 10,000, it's now 25,000. So you simply add more time to the chart to allow the evolution to occur when you see a problem. So more time seems to be the solution to so many problems that they encounter.
But then adding more time just makes the story that much more unfeasible. Because
Right.
again, like you pointed out, where's all the dead bodies? Where's the campsites, the grave sites? Where's the evidence for the story that they tell us? Okay, I think we did good on the topic of mutations. So the next question I want to ask you, Jerry, again, excellent book you wrote here. I recommend everybody pick it up, add it to their collection. It's a must read. The 3 Pillars of Evolution, Demolished. What are the 3 pillars of evolution, Jerry?
Well, number 1, mutations, which I cover in detail in the book. Number 2, abiogenesis, which also I cover, although there are some excellent books that show abiogenesis is impossible. And the third, of course, is natural selection. And natural selection isn't the problem. Natural selection basically reduces de-evolution. And so organisms that are not quite as fit as others are less likely to survive. And so actually, natural selection has a function against evolution because it reduces not only de-evolution, but also reduces problems that are going to build up because of mutations. So it does have a very important function. That's not denied. But and of course, Darwin's main contribution, they claim, was natural selection, because he didn't have a clue as to the origin of genetic variety. Right. And he had to come up with his solution was, we just have a lot of huge amount of normal variation. And so he felt the normal variation would at least partly solve the problem. Like with pigeons, a lot of normal variation, you can breed pigeons for the next 500 years and end up with a huge amount of variety without any new mutations. And he felt this is a natural amount of variety that we find. And he felt that natural amount of variety would be found in all organisms. Well, it's found in some dogs, cats, horses, but many animals, there's not much variety at all. Pretty much every squirrel is a few variations, color, but pretty much every squirrel is like every other squirrel. Raccoon, pretty much every raccoon is very much like other raccoons. And that's true with most animals. There's not that much potential for variety. Some, but not nearly as much as there is for dogs, cats, and horses, which in this case, there's a huge amount of variety for dogs, cats, and horses.
Right. What's interesting is the events, the speciation events that we observe, Jerry, on a genetic level, we see that they're typically due to a reduction in allelic variability, the sorting out of genetic variability, pre-existing variability, and shifts in what's called heterozygosity to homozygosity. So a lesser state of diversity. Well, doesn't that make sense if the biblical creation model of ancestry is true, that God would have created the original kinds with a lot of heterozygosity, a lot of DNA diversity, and over time you get adaptation, you get variation, but as time goes on, you lose a lot of that variation, so these creatures hit walls, and you see limits there, limits to the change now.
Right. Which even we've done with horses. If you could get a horse to run, 0.5 miles faster, you could win a ton of money. It's just
Right.
we know we're pretty much at the possible limit. You're not going to go much beyond this limit.
It's like, okay, we would agree that your domestic dogs and your wolves and your dingoes, coyotes go back to a common archetype, we'll say. So that common canid archetype had all the necessary DNA differences front loaded at creation to explain the variations. But going from a wolf to a chihuahua, that's hitting a wall, is it not? That's a loss of, you might be able to get 2 wolves and eventually get a chihuahua, but can you take a bunch of chihuahuas and get a wolf? Or has that variability been lost?
Yeah, that's an interesting point, because my guess is if you tried to breed wolves from chihuahuas, I have a feeling that you'd never get very far.
Yeah.
And you've already bred one way, so you've reduced a lot of genes, and so you're not going to be able to go the other ways. Some cases you probably could, but breeding tends to go one way.
Yeah, it's a great point. Okay, my next question, based on your book, I like the way you lay out your book too, Jerry. So in the beginning part of your book, you have chapter summaries, which give you a teaser, right? So people can look to them, look at chapter 2, read what it's about. Chapter 5, for example, progressive or degenerative, the mutation problem, which we focused on. And so obviously it's good to read the book from beginning to end, but perhaps people want to look at the chapter summaries and think, chapter 5, I want to dig into that first, kind of thing. And so you do talk about abiogenesis, and therefore my question to you, has scientific research falsified abiogenesis, Jerry?
It does more and more. What happens is they come up with a theory, seems to work, seem to have some good points, and then they begin doing more experimental analysis of this theory. And as time goes on, they tend to find problems with it. And so that's one reason why so many leading abiogenesis experts looked at the origin of life from outer space. Well, of course, that just moves the problem somewhere else, doesn't solve it. It just moves it in another place, which, of course, we can't really study very carefully. And some feel that we just don't know what the answer is. But when we find out how abiogenesis could occur, we will think, gee, so simple. Why didn't we think of that? And I watch a lot of films on various topics, science films. And I have one on Mars, and talks about why they're going to Mars. They're going to Mars because, well, maybe abiogenesis occurred on Mars, and maybe therefore we can finally prove abiogenesis. But of course, in spite of spending billions of dollars, we haven't found any evidence at all of abiogenesis anywhere on Mars. What they're hoping is they'll find evidence of past life on Mars. But from what I've read, and friends of mine that work for NASA, what they feel is we're never going to find any evidence of life on Mars. It never occurred, it couldn't occur, and the conditions on Mars are far more hostile than on the Earth. So if we're going to find it, we'd find it on Earth and not on Mars. But I'm surprised they can spend all this billions of dollars. Of course, American
Right.
government especially, we just like to spend money. We seem to feel that there's an unlimited pot of money, and NASA's got huge amounts of money to explore whether there's evidence in the past of life on Mars.
Just print it and spend it like nothing, right?
Yep.
It kind of like the evolutionary logic. You lose it all, apparently you'll gain it all. Keep losing money and you'll get rich. Keep losing genetic information and you'll take your fish to fishermen. So on abiogenesis, Jerry, those are some great points. Definitely been falsified. There are so many two-way and even three-way chicken and egg problems. One I like to point out is the fact that we've got, what is it, 60 to 100 trillion cells in our bodies.
Yeah, depends on if you're, small, short. You have far
Right.
less than if you're big.
Yeah.
Fat.
Some might have a couple 100 trillion
That
cells.
number is difficult because it varies a lot. Depends upon your body morphology.
And so from my understanding, from my study, we actually accumulate roughly 1 million new DNA mistakes or lesions in a single cell every single day. And so that's a lot of DNA mistakes in every cell every day. But thankfully, thank God, we have these amazingly designed DNA repair enzymes that fix a lot of those, or else, our population would probably go extinct after a couple generations. We wouldn't be feeling too good. And of course, many are passed on, as you pointed out, through the germ cell lines, but many are fixed. But what's interesting and a problem for the evolutionists in abiogenesis is the fact that these DNA repair enzymes, Jerry, the information for them are encoded in the DNA. But the DNA requires them to repair these DNA lesions or breaks every single day. And so my question to the evolutionist is, what came first, the DNA, but you need the DNA repair enzymes to repair the DNA, or the DNA repair enzymes, but you need the DNA to encode for the DNA repair enzymes?
Yeah, the repair enzymes are a big problem for evolution because it gets in the way of change, which they're looking for. So that's a problem they have, which they don't really think about much.
This kind of complexity, these mutually interdependent systems, they demand a designer, don't they?
Right. Which is what was believed for 4,000, 5,000 years.
Is that why the Bible says the fool hath said in his heart there is no God?
That would be one explanation, yeah.
Because
now what I see as a problem is that we're so inundated. I watched, just figuring out this the other day. I tend to stay up late and I can't go to bed.
Too.
I just lay in bed. So what I do is I watch about an hour and a half every evening. I've been doing this for 30 years. So if you think about it, 365 hours of film. So probably 300 or 400 films I watch a year. So you multiply that by 10, that's 4,000 films. Multiply that, get 30 years. So you end up with a huge number of films that I've watched. And virtually all of them teach evolution. I just watched a film, an excellent film, by the way, on cats. And over and over, this evolved, that evolved. This cat evolved from this cat evolved. And over, they talk about evolution, but they never had any evidence in the film, none, to show that evolution actually occurred. And so when you hear this evolution over and over and over, especially if you do a lot of reading about science, which I do, or watch a lot of films on science, you accept this as true because it's just part of your world, and it's an important part of your world because it's just hammered into you every day for years. And therefore, especially if you're a student, you see this over and over and over and over again, you just accept it without thinking much about it. Whereas the difference, I guess, between me and many people is I think about it. I think about, indeed, do they really have any evidence in the film for the evolution of one cat into another cat? They hypothesize, they can speculate, but they have no real fossil or otherwise evidence or even knowledge of how this cat could evolve into another type of cat. And there are, by the way, about 500 different kinds of cats, which surprised me. Some of the cats are really weird looking.
And yet we see connectivity. We see a continuum in terms of reproductive capability within these felids, from your house cats to your lions, to your jaguars, to your cheetahs, your tigers. And this is a continuum that agrees or supports with all these felid species going back to a common archetype.
Yeah.
off of the arc that had the pre-existing variability for those variations. And so it's interesting you mentioned, like, for example, that movie, that documentary, and you see this all the time with documentaries, articles, videos, or just in school, your basic textbooks. Like, I'm thinking back to in high school and college, the evidence that was put forth as evidence for common descent for me. And it's not really the technical points that we've been discussing for the first hour, Jerry Bergman. What the points are, for example, these right here. So I'll move us into these lines of evidence as we've demolished abiogenesis, mutations. We've touched on quite a bit in terms of so-called beneficial mutations. We've discussed tetrachromacy. We've discussed and engaged nylon digestion, HIV resistance. So we've been thorough. But one of the most common arguments that you'll see everywhere, Jerry, is homology. And so I took this from a popular article on BioLogos, Theistic Evolutionist Ministry Organization. And their article is titled, "What is the Evidence for Evolution?" And the first one they point to, if I remember correctly, is homology. So they say forms and structures demonstrate the validity of evolution. Jerry, I'll read this and I'd like to get your thoughts. When we examine the bodies of today's animals in detail, so, you can look at bats, you can look at humans, you can look at whales, and you're going to find similarities in form and structure, right? Like, for example, your forelimbs is a popular one they'll look at. We find some remarkable similarities. For example, the skeletons of four-limbed creatures, what scientists call tetrapods, are only slight variations on the same body plan, Jerry. So as you know, they call this descent with modification. There's variations on that same body plan because these creatures split over time, common descent, and these structures, they vary. They modify over time due to mutation, environment, selection, so on and so forth. The bones are longer in some animals, and in others they are fused together, but they are arranged in the same pattern. And so basically their argument comes down to homologous patterns in the biological world. Jerry, are these patterns good evidence for common descent?
Well, they're good evidence, I think, of the fact that certain designs work really well. And we for many years we had a Jeep. We sold it, but we have a Ford dealer right in town, and so we'd bring the Jeep there, and they would fix it. And talking to the mechanics one day, and he said, basically they're all the same. Yeah, there's differences, and there's certain tools you have to work on a Jeep, but by and large, they're all pretty much the same. A car is a car, and if you have a sedan or convertible, there's so many similarities, and therefore the similarities are there because, indeed, there's certain things that work. And the best example of this, looking at cars again, is where cars used to be so different. The Volkswagen was 30 years ago was very different from so many of the cars that we see today. Well, now they all look pretty much the same. So a friend of mine just bought a new Porsche, and I expected to see something very different. He pulled it out, and it looks like a Ford. I mean, he spent all this money for a Porsche. A Porsche is basically looks very much like, it costs more, I guess, than a Ford, but it looks very much like a Ford. And of course, the reason is because people find as a whole that the not the sedan, but what do they call the cars, like a Jeep? But basically, they find those cars are convenient. You can have room in the back to store things. You can carry the family in the car, And therefore, virtually all cars are designed very similar because of this common demand, and this common need that people have, and common choices that people have. And therefore, you see a lot of common structures because is there a better way of designing it? Could you think of another way of designing dogs as opposed to the way they're designed now? No, you can't. And it's true, there's a lot of similarities, of course. Even the organs, lung is similar in virtually all terrestrial animals. Very similar in virtually all terrestrial animals. Why? Because that design works really well. Designs are very functional. So I would say homology is, I cover that in my second book. Homology is a very poor argument for evolution because homology is there because of structural limits and advantages. And you find the same thing is true in cars. Instead of the cars becoming more different, as you would expect, the cars are becoming more similar. And therefore, this, my friend's Porsche looks pretty much like my Jeep. And my Ford we have now looks pretty much like the Jeep. The design is very, very similar. There are differences, but by and large, it's pretty much the same. The same basic design is there. And that's why if you know how to work on one car, generally you can work on many other cars. And I noticed in the Ford dealer, they have many cars they work on because they're pretty much all the same. Doesn't always work that way, but by and large, you have a similar design because of similar requirements. So thus in tetrapods, you have a very similar design because you have similar requirements. And therefore, you're going to find the similarity throughout the animal world. But yet, on the other hand, you sure see a lot of differences. Differences that you have to wonder, is there, like penguins are so different from dogs. And so there are animals that have major differences, but of course, a penguin lives in a different environment than a dog does. And therefore, animals that live in a terrestrial environment or breathe air, you're going to find a lot of similarities. And that may be one reason why when you transplant organs, we find a number like the valves in a pig are transplanted in humans because, well, heart in pig does pretty much the same as in humans. And for several reasons, the heart in a pig is more advantageous to use in a human than hearts from other animals. But nonetheless, you see a huge amount of similarity. That's why when I taught anatomy, the college, we didn't have cadavers, and therefore we dissected other animals because, again, there are differences, but basically the design of all terrestrial air-breathing animals is pretty much the same.
That's right. You could take automobiles built in North America, Asia, and in Europe, and you're going to find all kinds of similarities. You're going to find homologous patterns. That doesn't mean that they all share a common ancestor in the Arctic somewhere. No, it's good design. Jerry, I think you nailed it. I like to say the Bible is clear. We as humans are made in the image of God, and therefore there must be something about us that reflects the divine. And so perhaps we can get an idea or a sense into how God designed the biological world, Jerry, based on the way we as human engineers design things. And it just so turns out that we design in homologous patterns. God designed the world in homologous patterns. We even design in nested hierarchical patterns, where sedans look a lot more similar to a hatchback than they do to a boat or an airplane. So you have these natural hierarchies that are reflective of design and not descent.
Yeah. To me, they're scraping the bottom of the barrel when they try to use homology to prove evolution.
Their follow-up, Jerry, is typically this. Well, Jerry, cars don't reproduce. Biological organisms do, and so it's a false analogy. What's a good response to that?
Well, that's true. Animals do reproduce, and we see a lot of similarity. But on the other hand, we see a lot of, like penguins. Penguins are just so different from any other animal. How about ostriches? They're so different from any other animal. And so there's a lot of similarity, of course, but there are animals that are so different, evolutionists have a hard time explaining them, like dinosaurs and birds. Huge difference. So yeah, you see similarity, but saying something from a similarity is problematic when you see so many differences. We as creationists look at the differences and say, wait a minute, why would this particular animal be created to be so different from some other animal? I think I like the idea that God created life in such a way so that it does not look like it evolved. So he created life specifically to look like it was created and did not evolve. And that's called the biotic message. And so God is telling us something when we look at life. And if it evolved, I would expect should be a lot more similarities.
Yes. God made it clear for many reasons, the complexity, the mutually interdependent systems we talked about earlier, that life did not evolve from inadequacy to precision. And so, Jerry, the next argument they'll use, and I like to say when they say, well, cars don't reproduce, I like to say adding reproduction adds more complexity. And so it doesn't help the evolutionists, Jerry. It actually makes the problem worse for them. God is so far ahead of us. He's designed and created his creation, the biological order, with the ability to reproduce and pass on their genes. Imagine we could do that. Imagine car manufacturers could design their automobiles to just replicate. How much money would they save on workers? How much money would they save on production? If we could, we would. But we're not that
designed to repair themselves.
So the fact that God created us to vary and change and pass on our genes, that speaks to his knowledge, his infinite capability of design. And so the next argument that they use, Jerry, has to do with homology, but now on a DNA level. So we look in the genetic sequences of different species or creatures in the biological world, and we find homologous patterns, just like we do with the morphology and anatomy we're speaking to. But now we see it at, I guess we could call it the blueprint level. What would be a good response?
Well, of course, if you have homology in the morphological level, you'd have to have a lot of homology in the genetic level. And when there's a lot of homology, but I was amazed in discovering the fact that if you have a large area with 45 trees, and you have this murder case I'm thinking about, and a car brushed across one of them, okay, could you prove that this car brushed across this specific tree among hundreds of others, not only in this one area, but in other areas? Well, they found genetic analysis, they can. And so there's been a person convicted of murder because they found he brushed against a part of a plant, and they proved that the plant he brushed against specifically was this plant, which they found in this area. And the other side argued, well, wait a minute, this doesn't prove anything because they're all the same. The DNA of the plants are all the same. They found, no, there isn't. Just like people are all different. You can distinguish everybody in the world by their DNA. Likewise, you can, I assume, at least in this one area, you can distinguish every plant in this area by their DNA. So when you do DNA analysis, you know that this car brushed against this specific plant, which is right here, right near the location of the dead body, period. So this place is the car right near the dead body, period. And so that was the main argument they used, which, by the way, the court accepted. And the other side argued that, wait a minute, maybe he just brushed against some other tree over somewhere else, some other park somewhere else. And they proved that he didn't do that. He brushed against this specific tree, which was right near the body, which they found. And that surprised me that the DNA would be that different. I just assumed, I guess, until I watched this show, that the DNA in all trees would be pretty much the same.
Right.
Like people, it's not. And therefore, and they've done this now with dogs, with animals, and so on. And so we know that instead of being identical, we find the DNA of humans, dogs, animals, and so many other, in this case, plants, is different. Why would all the DNA be different among all these plants? I mean, to me, that's the baffling thing, but yet it is different, which is convenient for the forensic analysis because they proved this person was guilty, and they found guilty and based on this plant DNA.
I like to say It's the differences that make all the difference. Creationists, we can explain the similarities and the differences. We can explain both homology and homoplasy, where maybe you could speak to this. So anytime there's a discrepancy in the universal phylogenetic tree of life, Jerry, what do evolutionists invoke? Convergent evolution. Well, these are similar traits. We see similarities, let's say in echolocation or the eye system, something like this. But there is no recent ancestry with these creatures. A big one would be a dolphin, ichthyosaur, and a shark, where they have a lot of similarities, streamlined bodies, designed for the ocean environment. But according to the evolutionists, they don't share recent ancestry. So what do they result rather than homology? Or what do they invoke? Homoplasy or convergent evolution. The, the independent evolution or acquisition of these traits over time. Isn't that just a rescue device? Can you speak to that a little bit, Jerry? Homoplasy and convergent evolution?
Yeah, it is a rescue device. It just reminded me of a quizzes given to children. And the quiz was they had a triangle, and they wanted the students to calculate the hypothesis, if I pronounce it right. It's been a long time since I taught geometry, but the longest line. And there's an X there.
And Jerry, I just wanted to jump in real quick. Feel free to take your time on this one. I just have to fill up my water real quick.
Oh, okay. But the question was, calculate X. So he basically, instead of calculating the hypotenuse of the triangle, he calculated the value of X, which was not hypotenuse, what the teacher wanted, but he circled the X and then said that this is the value. And he didn't calculate the numbers, which is what the teacher wanted. And there are so many of these examples. And so it illustrates, I think, quite well the fact that if you, in this case, the point was if the student doesn't know the answer, he can come up with an answer, which I should have saved some of these. They're really cute, but illustrates quite effectively. You can answer it and it seems right, but it's not what the teacher wanted. And same thing is true in the whole evolution issue. You can come up with an answer which sounds right, but it's not right. And you see that so often. And yes, I should have copied some of these off. They're really cute. And how these kids answer the question. And so it's a good example of the fact that you you come up with reasons. I taught forensics for a while, and it's amazing how people can come up with reasons which they think will explain the problem, and it doesn't in the long run. But in many, get something in my throat here. But in many ways, what they're doing is they're coming up with reasons which, on the surface, appear valid, but which in fact are not at all, and not valid at all. And it's a rescue device, as you mentioned, and it explains it, but it doesn't really prove it.
Yeah, and it's imagination. Evolutionists hope, dream, and imagine that the sophisticated eye system could evolve 30, 40, 50 times. The sophisticated echolocation system of creatures like dolphins, whales, bats could evolve independently several times. It really is science fiction. And the pervasive pattern of homoplasy it undermines their whole argument from homology, because the homology on its own for the evolutionists to invoke, or for the evolutionists to say, well, this is only evidence for common descent, that's affirming the consequent. Well, perhaps there's another explanation, evolutionists. How about common design? If I walked outside and my grass was wet, I could conclude it's because it rained out. Well, maybe there's another reason. Maybe somebody had their sprinkler on. So I think they're guilty of a logical fallacy on that one, and many of their other points, Jerry.
Yeah, I think I covered that in my second book, but yeah, that's always been a problem. for evolutionists, and they just say,
Right.
well, coevolutionists, or this explanation they come up with are really problematic.
And I like to say to their assertions, prove it. They're going to say, well, the sophisticated eye system, the camera eye, must have evolved independently many times. And that's due to convergent evolution. But that's assuming evolution to prove evolution. And they're just making that assertion without any real evidence. And so I like your point. Yeah, the blueprint has to share similarities if the structure and the form also has similarities. You're probably going to find many similarities in the blueprint of various modes of transportation designed by GM. Isn't that right, Jerry?
Yeah, like why are all tires round?
Yeah.
Every tire I've ever seen is round.
Yeah.
The only design that works. And so many designs in cars and people are that way because it works.
One pervasive pattern of homology in the biological world, to me, Jerry, points to the God of the Bible, because the God of the Bible is one God, monotheistic. And so one overall general pattern points to that one God. If there were multiple types of patterns, different DNA blocks, rather than RNA, protein, and DNA for some creatures and not for others, that could mislead us into believing God is polytheistic, multiple gods created creatures. No, one pattern suggests one God, in my opinion.
Yeah, one designer. Yeah.
Okay, so the next point they'll make, Jerry, and this is right from the BioLogos website. So these guys, they're really trying to push evolution, right? They like to say, well, you don't have to believe in young earth creation. There's alternatives. And so they will look to vestigial traits. And I had a slide on it, but as we were talking about earlier, we have so many slides, I might not be able to find it in time. And so basically they'll say the vestigial traits that we find. Oh, here it is. Vestigial structures. So they actually follow up their homology with this. So they'll say, this is from the article, we can also look at the bodies of animals today and find features that are similar to what other animals have, but which no longer seem to function or have different. notice how they've revised history. They revised their definition, Jerry, because now we find that all of these structures and organs have essential functions. So now they've redefined vestigial trait as meaning not necessarily having no function, but maybe they've altered their function or have a different function. So they put that in brackets. Anyway, so they say scientists call these vestigial traits. What's a good response to this argument, Jerry?
Well, that surprised me. I taught anatomy for, I don't know, 20 years, and I used a number of different textbooks, and I reviewed a number of textbooks. And one thing I always looked for was in the index, the word vestigial. It wasn't in any of them. And when I looked at the so-called vestigial organs, the appendix, for example, none of them mentioned that any of these structures were vestigial. The section on the appendix talks about the 5 different functions of the appendix. This section on the os coccyx talked about the different functions for the os coccyx. None of the arguments that they commonly use for a vestigial organ were talked about as being vestigial. They were all talked about as their function in humans. And therefore, I just find intriguing. And yet, if you look at the biology books published by the same publisher, there typically is a chapter on vestigial organs, or at least a section on vestigial organs. So why the anatomists agree that there's no vestigial organs, not in the book. If there was, they would include them. And none of the books had any reference to vestigial. And they all had the explanation of what we now realize they are for. So why is the anatomy book so consistent? They're not there. And the biology books, why are they so consistent in claiming the vestigial organ idea? And I just found that intriguing when I reviewed textbooks. Typically, each year you get 3 or 4 new anatomy books, and you review them to see whether or not you want to use the same book again next year. And so I got a whole box full of several boxes full of anatomy books. So among anatomists, the vestigial idea, from what I know, is not there. You talk about vestigial organs, anatomists will look at you wondering, what planet are you from? Clearly, all these structures have important functions, and they don't talk about vestigial functions at all. They talk about the uses of all of these structures in humans today.
So, if anything, evolution hinders scientific advancement because we expect to find treasure, they expect to find junk and evolutionary leftovers.
There's a lot of organs that were claimed to be vestigial, but were found to be not only not vestigial, but critical for good health. The thymus, for example, the pineal gland, the pituitary gland. So many of these believed to be vestigial, we now know are not only not vestigial, but very, very important. And that was, one of them was discovered by a doctor who they used to remove parts, I think, of the thymus, and they noticed that they ended up developing some problems as a result of this. In one case, they were twins, and the one twin, the entire thymus was removed, and the other twin, only part of it was. And they noticed in time how they developed very differently. And they concluded from this, which now we know is true, that these structures are really very important. You cannot remove them without a problem in the long run.
I've heard them as a counter rebuttal. They'll say, well, the reason why we know or can say that these vestigial organs or structures have simply acquired a new function or changed its original function is because, and they'll point to an example like the appendix. They'll say the appendix has a different function in different creatures. So depending on what you're looking for, maybe a carnivore or herbivore or a human and a chimpanzee, sure, they admit that the appendix has a function, but it's simply acquired a new function, or else the function would be the same in every creature. What's a good response to that?
Well, that's again another explanation they're trying to use to explain it away. It reminds me of my cat film I watched, where they'll show a cat that survives so well in Antarctica. Cat survives really well. And they'll say, well, this cat was eventually evolved into this cat we see now from this other cat. But of course, they had no evidence for that. They had no evidence at all. But they did, of course, show quite well that there's some cats that have such an effective fat and fur covering that in the Antarctica, really, really cold, 20 degrees below zero, they have to find places to cool off. They get so warm. And they'll find a log that's covered with snow, for example, and they'll lay on the log to help cool off because they're so effective in being able to retain heat in these very, very cold areas. And they say, well, they evolved from this other cat from Africa, but of course they don't know that. That's just an assumption. But they know they're very, very different, and you have to wonder how this cat from Africa survived in this 20 degree below zero temperature weather in Antarctica. How could they survive? Well, they don't know. But that's what they hypothesize. They have to come up with an evolutionary explanation. And that's a good example, which I guess if you watch that, it would seem to really prove evolution. But in fact, and of course I can look at this critically, but in fact, it did not at all in any single case prove evolution from one cat to another cat. They just hypothesized that this cat evolved into this other cat. And that's the end of their claim.
A lot of storytelling. Variation in designs in the biological world is, again, what I'd expect. Human engineers design variations as well. I mean, you can get a Volkswagen Jetta with a diesel. I mean, maybe not anymore, or with a regular engine. You can get your car with a whole variety of steering. How come here in North America, we drive on the left side of the road, but in Britain, you drive on the right side of the road? Same function, same vehicle, just variations in that. And so this evolutionist in the chat, he's been very energetic, bringing up a lot of these arguments that we've addressed, but he's pretty adamant about them, and he wants us to address them. So let's address these all, leave no stone unturned, and he's put these all here for us. So he says, we have loads of evidence. We've observed evolution via genetic mutations. Well, you started off the show, Jerry, for 30 minutes or so, showing why no, mutations are not going to help take your fish to fishermen. They're not going to help explain the necessary vertical changes that evolutionists would need. They're largely deleterious. Most are nearly neutral. They build up, they degenerate. Gene duplication. Okay, fair enough. We haven't talked about that one today, and that is on my list. So gene duplication, Jerry, they'll say, well, you could have a region of the genome or a sequence. You can have a large gene duplication, a smaller gene duplication, and that duplicated region, they call it neo-functionalization. They'll say that duplicated region can now experience novel mutations, beneficial mutations, and thus altering it from its original sequence and evolve a new function. What's a good response to that?
The main problem with that is that when you have genes that duplicate, and of course that does happen at times, certain diseases are a common problem is due to gene duplication. But when you have that occur, what we see is that the hotspots in one gene, it duplicates, the same hotspots are often found in the other gene. And so therefore you have an equal likelihood of this damage in one gene as well as the other gene. The claim is that if you have a gene duplication, the one gene can function, do what it's supposed to, the other gene can mutate and do something else in time. Well, that doesn't work. There have been no clear examples of that. In fact, what we know is, in general, when this occurs, it causes a problem. It doesn't solve any problem. And that's partially because mutations are just as likely in the duplicated gene as in the other gene. And that's what often happens. Not always, but that's what often happens. When you talk about, by the way, the fossil record, I spent a lot of time looking at the fossil record. And a whole book on that, fossil forensics, which clearly shows we don't have evidence in the fossil record of any evolvement of any organism, any bears, etc. Bears, penguins, etc. The fossil record is very clear, especially with insects that are trapped in amber. We see no evidence of evolution at all. And if you look at, I have a book called The Evolution of Insects. Very clear, it states we have no evidence for the evolution of insects. We have a lot of beliefs about them, we have theories about them, but there's no clear empirical evidence of the evolution of insects. And they not only look at amber cases, trapped in amber, but they also look at other examples like the fossils between rocks, in which the insect forms a pattern on one rock when it's split. The three examples they use over and over again are whales, horses, and whales, horses, and what's the other one? I can't remember the other one. But I've examined all three of these very carefully and find the evidence for evolution of whales is pathetic. All they're doing is guessing. They find animals which have structural similarities to whale, and therefore they assume this is a transitional form. But it's not. Same with horses, which I worked quite a bit on. That was the classic example of evolution for a century. And all we find, and this is pretty well agreed among the experts in this area, not just me claiming this, but pretty clear that what we have is not evolution, but we have a lot of variety of horses. And now these evolutionary trees they proposed, those are all been thrown out. Basically, I can talk here. I think 2 hours is my throat kiss. Anyways, if you look at the fossil record of horses, all you find is a great deal of variety in horses. You don't find that nice, neat progression that has been pictured in the biology books for the last 150 years. It's not there. And they recognize that. They recognize that what we see is not a progression as we expect, but what we see is a wide variety, an enormously wide variety of horses, which supports the creation world. And you can see horses, whales. I don't think of the other one now. It's been a long night.
You've done an excellent job, Jerry.
But anyway, I looked in detail the three best examples. And so there's a single good example we have of the evolution based on the fossil record or based on evidence of the evolution of any animals. You can line them up. That doesn't prove one animal evolved from another animal, as we now recognize with horses. Oh, humans. Third is humans. Humans, horses, and whales.
Right.
And humans, I've very effectively refuted more and more you see the same thing with humans. We're not talking about the progression from an ape to a man. We're talking about a lot of variety in the fossil record, which doesn't show progression. It just shows a lot of variety. And that more and more is being recognized. In fact, more and more paleontologists are throwing out the whole progression idea. It's not true. In essence, they're throwing out evolution. They're saying that what we have is a lot of variety in primates, and that's all we're finding is variety. We don't have a progression from an ape or common ancestor to modern humans.
I love that you killed three birds with one stone, because those were on my list. And excellent endurance tonight, Jerry. This really is. I thought maybe I'm being too ambitious, trying to basically address all their best lines of evidence in one show, but we've done well. And you just encountered whale evolution, you engaged human evolution there, and then also what was the third one? Getting late for me too. Horses, yeah. So basically, what we see in terms of those is simply like what they claim are transitional forms. The ones they put forth can easily fit within the variation that we've discussed in biblical kinds. We find a genetic continuum between various animals, right? Cats, dogs, horses. And we just see a reflection of pre and post flood diversity. But the same gentleman in the chat, I pointed out that the Cambrian explosion is a problem for him. And he keeps pressing me to say why. Well, here's the thing. In the Cambrian explosion, there's this sudden appearance of all these major phyla. Jellyfish, coral, trilobite with their sophisticated eye system. They just pop up out of nowhere. And these are massively different kinds of organisms. These are phylum level differences with no evidence at all of evolutionary change. So my challenge to the evolutionists is, where's the precursors? Where's the ancestors that these groups of creatures, these highly complex and sophisticated creatures, where are their ancestors? Where are their preforms? They don't exist. They just suddenly appear. And that's what we would expect as creationists. Isn't that right, Jerry?
Right, right. Of course, they're coming back as well. Just keep digging and looking. We'll find them. Someday they're there. We just have to find them.
Exactly. Yep. They're there, or they'll resort to the whole fossilization is rare, or soft-bodied organisms can't be fossilized. But correct me if I'm wrong. I mean, we have fossilized jellyfish. We even have fossilized bacteria, don't we?
Yeah, there's a lot of fossilization has occurred, which preserves these organisms in pretty good shape. It's surprising how well some are preserved, especially, of course, amber. But there are other conditions, tar. There are other conditions which preserves fossils quite effectively. So it's not just amber, but we have millions, of course, examples in amber. And I should mention that there are, 13 billion fossils? There are so many that we have not explored or analyzed them very well. In fact, every now and then you find a fossil which has been in the collection for 100 years. Finally, some student's trying to get his degree, and so he analyzes this fossil who's been there for 100 years. The reason that there's so many fossils, where do you start?
Right.
Museums, some have huge rooms with drawer after drawer after drawer of thousands and thousands of fossils. I think one estimate is there are 10 million fossils or parts of animals out there. So there's a huge fossil collection to be able to do analysis.
And yet what we find is sudden appearance, stasis, and extinction. And a few interesting candidates that they like to push over and over again as so-called transitional fossils. I think we can debunk every single one of them, as you have, Jerry. You and I, we did a whale evolution section in a video because I see a lot of commenters bringing up the usual candidates for whale evolution: Ambulocetus, Durodon, Pakicetus, Basilosaurus. But I want to refer people to that video. I've got it up on the channel. I've titled it something like Whale Evolution Debunked. It's about 45 minutes, and we go through every single one of those examples.
Who did you interview for that?
Oh, no. So you and I, we did a program together, and in the discussion period, we touched on the examples. But actually, I clipped that together with others. So I also had several other programs with Carl Werner, who did the series on living fossils. And he has some, in his documentary, he's got some great interviews directly with the whale evolution proponents, like Philip Gingrich, one of them, where they make some really interesting admissions in terms of the lack of evidence for whale evolution. So I put that together in a video that I'll link to people. And okay, you kind of just debunked this, Jerry, but as you can see here in his comment, he says, what about predictions made in the fossil record? And so it's kind of a general statement, isn't it? I'd say prove it. But just from what I've heard from other evolutionists, they'll say things like, well, we predicted a transitional form between, farm, fish, amphibians, reptile, mammal. And so when they found Tiktaalik, they call it the famous fishapod. They say they found it where they expected. And so they predicted it, they discovered it, and therefore that verifies evolution. What's a good response?
I'm not too impressed with Tiktaalik. I think it's pretty bad example of a vestigial organ. And I think when you talk about forensics, if you would argue that in the court of law, I don't think you'd win the case. They would see that too many differences. And of course, there's so many fossils out there. I used to take my students on fossil hunts, and there's certain places in Sylvania, actually, which is near Toledo, where I bring my students and they do fossil explorations. And they almost always, they would come home with a box of fossils they found. There are lots of fossils out there, lots of, especially clams and sea organisms. And every now and then they find a trilobite that's worth lots of money. So I know at that place in Sylvania, they found a trilobite that's worth a lot of money. And so if you make enough predictions and do enough digging, I imagine a few of them will seem to appear to come true. It's like so many predictions. We make a prediction and then it seems to come true later on. And it's actually, kind of distortion of what we said that came true. And so I don't buy that. They claim relative to Tiktaalik, but that's very unimpressive. In fact, the Gingrich and others are having a hard time convincing the evolutionists about the validity of that find and what it means.
Yeah, and they don't like when I say this, but it's because it's an argument against their so-called prediction, and I'll keep using it. They discovered tetrapod foot tracks in Poland. After discovering Tiktaalik, years later, they found true tetrapod foot tracks in Poland, predating Tiktaalik, according to their evolutionary timeline, by about 20 million years. So how is that a confirmed prediction? I mean, so they're going to have to say, well, this is just a side branch or an example of what that transition looked like. But even according to your model, 20 million years before Tiktaalik came about, you had true tetrapods existing, leaving their footprints.
If you make enough predictions, some are bound to come true. And make them vague. I have friends who like to study people who are doing things like predicting the future and people who are fortune tellers and so on. And they discovered how they do it. The predictions are vague enough where it's bound to be fulfilled because a lot of events can actually fulfill it. And they, in the books that I've read, which is not a major study of mine, but people can get pretty good at making predictions, and they seem to come true when the predictions are made with enough leeway, enough generalities that they can extrapolate what happened to those predictions. They can say, well, I predicted this and it happened. See, there we go. And so, and again, you predict enough, eventually you'll find some fulfillment in some way.
Right. The kind of predictions that they make are like, okay, they're going to watch me swing a baseball bat and hit a ball 200 times. So they're going to be able to see what my average is. And then they're going to predict, oh, he can hit it 250 feet. It's not that impressive. They've already watched me hit the ball 250 times. Now, if I were to just point to some random guy off the street and said, hey, I predict you're going to hit it this far, and he does, now that's a pretty specific prediction, but that's not something evolutionists can do, eh, Jerry?
Yeah, that's true. It's not. And again, I'm not very impressed with that Tiktaalik.
Yeah, me neither. Okay, we're going to start winding it down, Jerry. This has been great. Oh, but with your gene duplication answer, excellent answer. If I handed you a copy of your book, so you own this book, you wrote it. If I handed you another copy of it, are you going to learn anything more by having a second copy? No, it's the same book, but now you have two of them. And then if I started introducing typographical errors into your second copy, it's not going to make for a better book. You're just going to have, as a matter of fact, you're going to have a worse book. It's not going to be as good as the original. That's like their duplication argument.
In the medical field, gene duplication in general is a problem, especially with certain, like Duchenne's muscular dystrophy can be a problem. And there are a number of diseases where it is a serious problem, gene duplication, because a few extra genes can be beneficial, but beyond a certain point, you have a problem. I'm trying to think of the disease that is referring to. It's a neuromuscular disease that causes you to go.
Huntington's disease?
Huntington's disease. Huntington's chorea. There you go. Your medical background, you can pick up some of these. Yeah, I studied.
Yeah. We got that in common, brother.
For a while, but I haven't thought about it for a long time. But yeah, Huntington's disease, when the duplication is at beyond a certain level, then you have a problem.
Yeah, you're exactly right. Thousands and thousands of mutation-related diseases, and they can barely point us to a truly beneficial one that we won't. You just debate them all day long. You know, there should be no question. People asking about the whale evolution. So I did post the video that I recommended people watch in the chat. Actually, Doki, our chat mod, did. And here's some slides that we covered. Right. So here's all your examples, and you can see that they can be split up into fully aquatic, primarily terrestrial, and fully terrestrial. So there's no real transitional forms. And I find it interesting, Jerry. I'm not sure if you can see this slide here, but this is their wild imagination. This is Pakicetus here, because they believe that lineage is basically Pakicetus, Ambulocetus. Ambulocetus was very similar to a crocodile, but like a mammal version. So just an interesting extinct creature, not evolving into anything fully aquatic. And this is the end of the line, a blue whale. Is that not a wild imagination, Jerry? This little Pakicetus, wolf-like creature, to a blue whale? Those are some serious changes.
Exactly, enormous changes. And I cover that in my article on the whales. That's true there.
I'm going to post your article because you do write you wrote an excellent article, and I have your book behind me, brother.
Can you send me your PowerPoint? Some of those I would find very useful in my.
Sure. Yeah, absolutely.
Steal a couple of slides. You can send me that. I think I guess you could, if it's more than 25 megabytes, you can't email it. You'll have to.
I can probably send it over Google Drive, because I do.
Google Drive. Okay.
When people ask for slides, and I'm always adding to them as well. So I'll send you, and I got some good ones on genetic entropy in the chromosome 2 fusion. This one I find very interesting. This is from Philip Gingrich himself, who's an evolutionist who works on this. He said this, Jerry: Ambulocetus has its eyes raised up on top of its head in a very strange way, and it is unusually large for an early whale. Maybe it's not the main line of evolution.
Yeah.
That's a famous one they've been using for a while, and now that's even being questioned. So, like you said, Jerry, I'm not impressed with their best so-called examples. I mean, Philip Gingrich right here said, I speculated that it might have had a fluke, Rhodocetus. I now doubt that Rhodocetus would have had a fluke. So fossils are fickle, and there's so many of them that you can tell really any story that you want. Isn't that right, Jerry?
That's true.
Okay, Jerry. So here's the last one we have for you, and then we're going to let you get out of here for the night. I really, really appreciate your time here. I'm sweating trying to get through all these, but we did it, brother. I think this was a really good, all-encompassing show for people. We've had a lot of audience interaction, Jerry. So you're what we call here a fan favorite. You're always a blessing. So biologos, it's basically the same argument that you just demolished, but now they apply it to the genome. They call it genetic scars. So this is right from biologos, Jerry. So they say just as scars stay on our bodies, right? The vestigial traits that you just debunked. Just as scars stay on our bodies as reminders of past events, the DNA code also contains scars, and these are passed on from generation to generation. They say DNA scars result from the deletion or insertion of a block of bases. I want to say this, Jerry, before you respond. There's two very different explanations from the evolutionists and the creationists when it comes to the origin of genetic diversity. Evolutionists, they explain all genetic variation as being the result of mutations over time. So they look to DNA differences and they explain those as mutations. But we as creationists, we look to the vast majority of DNA variation, specifically autosomal nuclear DNA variation, as being the result of initial design, created heterozygosity. So where they say something like a deletion or a substitution or an insertion, they're simply inferring based on their worldview presuppositions that these are a mutation. So I do want to make that clear to any evolutionists in the chat. Just because you say deletion or just because you say mutation does not necessarily mean that that's a mutation. Or if they look at a spot in the human genome, Jerry, where there's a DNA unit missing, but it's in the chimpanzee line, well, they'll say, well, the reason why it's missing is because either it was added or it was deleted, or maybe it was never there. They won't even question that one. So anyways, they say because we have a lot of these, hundreds of thousands, and they can be precisely located, they serve as a historical record of species. So basically they say we share these genetic scars or genetic mistakes with chimpanzees, gorillas, orangutans, also known as pseudogenes. And so we must have inherited those from common ancestors. What's a good response, Jerry?
Well, what we find is over and over these so-called pseudogenes, as well as some of these others, are simply a design function which has a purpose. And it's like labeling things vestigial. We label them vestigial, and then later on we find they have a function, a use. And I think a lot of these so-called damage is, and it could be damage, that's not the debate, it could be damage. I mean, we accept the accumulation of mutations has been occurring since Adam and Eve. So therefore there is damage to it. But on the other hand, you got to be careful in assuming this is damage when it indeed does have an important function. And the lesson for that is what we know about vestigial organs. We've started out with 100 around the turn of last century, and now we have 0. So therefore, from 100 to 0 through more knowledge. And so my supposition is, as we get more knowledge about these so-called damaged genes, we may change our mind. They may not be damaged. It's a judgment someone's making. And there could be some. I mean, we don't deny there's some damaged genes. After all, we have 6,000 years of mutations. So there's some that are there. But on the other hand, got to be careful. They may not be all, or most of them may not be damaged. Most of them may be a result of design, which appears to be damaged.
You nailed it. And that is oftentimes put forth by more informed PhD evolutionists as one of their best lines of evidence for evolution. And no, we're finding that a lot of these so-called genetic scars are not genetic scars. William Lane Craig, well-known theistic evolutionist, he's great at some things, but he's wrong on evolution. He looks to pseudogenes as well. So it needs to be countered, and you did so. We've documented the fact that many of these are necessary to sustain healthy life processes. Many evolutionists have said, well, why do the pseudogenes look like their healthy counterparts, Jerry? But I could pull up at least 2 papers that demonstrate that the pseudogene expression actually helps to regulate the translation of its protein coding counterpart. And therefore, it has to look like its counterpart to be able to help with that function.
That's a good example, a good point.
So there you go. Pseudogenes debunked. Same thing goes with all of your other so-called junk DNA. We know there's a lot of functions that they did not expect or predict. ERVs, functional in gene expression, embryological development, functional in the immune system, tumor suppression. So there's ERVs, pseudogenes, loose sequences, same thing, junk DNA in general. So, you know, there's a quick, in a nutshell, debunking of those points. So, Jerry, excellent show. I'm sweating, but we did it, brother.
Good. Thanks.
Yeah, I think we addressed your major and minor points for evolution. We've still got a great chat. We could go for another 10 hours, I'm sure, but we do got to wrap it up at some point for sake of our health and voices. So, Jerry, let me hand it to you. Actually, before I do want to recommend people check out your book. It's a must-have, The 3 Pillars of Evolution, Demolished. I've got your books behind me. C.S. Lewis, was he a Darwinist? The answer is no. I've got your book, Fossil Forensics. Excellent book. So I do want to encourage people to support you. You're doing great work. You've put out several articles recently, which I didn't really have time to get to in terms of questions. So I'll ask you those our next time together. And people can check out those articles. I've linked the website in the description box for people to see more from you. They can also go to standingfortruthministries.com. Go to Guests on Standing for Truth. Scroll down to Jerry Bergman. I'll have to update it with our last couple programs together, and they can see all our past programs and podcasts that we've done. So, Jerry, I want to hand it to you, brother. Again, I really appreciate your time. I know how busy you are. I really appreciate the fact that you're always staying up to date on these points. You're always doing your best to counter the critics, to counter the skeptics and their best arguments. It's very important, I think, as creationists that we do that. So let's hand it to you for some final words and final thoughts for tonight.
Yeah, well, it's good to be on here, getting the word out. My main concern is some of the evolutionists don't read what we have to say. Because a good example is when I worked at the college, I did a paper on the evidence for evolution of snakes. And I found just by consulting the literature, there is no evidence for the evolution of snakes. There's some theories, there's some claims relative to loss of certain feet structures. But on the other hand, there's no good evidence. Well, if one of my colleagues was getting his doctorate in herpetology, studying snakes, and I thought it'd be a good idea to give it to him because I don't have a doctorate in herpetology. That's his field. So I wanted to give him the article to look it over, and maybe I made some mistakes. And he said, nope, he refuses to read it because I'm wrong. I said, well, could you read it? And just check my references. It won't take you, but it's 15 pages. It's a summary. Nope, I'm not going to read it because you're wrong. And I can guess that this teacher now teaching, he's at another school now, but I can guess that when he covers evolution, he probably talks about the evolution of snakes, unaware of my work. And again, I quoted from leading authorities on snakes. I didn't, I'm not a herpetologist. I just looked at the literature and looked at books on snakes, and many had, in fact, some had large amounts of information on ideas about the evolution of snakes. But basically, there's no evidence, and they admit, herpetologists who are in this area admit that there's no evidence for the evolution of snakes. And he just wouldn't read it. And I have a feeling so often that is true with so much of what we produce. They just don't read it. They're unaware of it. And therefore, they accept ideas, which if they read what I wrote, my guess and my hope is when he taught the evolution section of his course, he probably would say, well, we have no evidence for the evolution of snakes, but we have plenty of other evidence. So there's no, need to worry. But on the other hand, he's a colleague, he's a friend. Him and his wife, his wife and I, we had offices right next to each other, and we knew each other pretty well. Just for courtesy, just read the paper, 15 pages. It was his field with snakes.
Right.
I'm sure he could find something, mistakes that wasn't clear, but nope, he wouldn't read it. And that's disappointing.
It's because they want to exist in their echo chamber, and they want to keep going back to the drawing board, as we've demonstrated today when you point out the errors or lack of evidence. You mentioned herpetology and snakes. So I'm just curious, do you believe, because we've had in the past an evolutionist herpetologist on for several debates, and he would point to fossil snakes. One, if memory serves me well, is called Najash. And he'll say these here in the fossils are evidence that snakes once had legs, hind
Yeah,
legs?
that's been refuted by the herpetologists. Basically, those are copulatory organs. As you can imagine, for two snakes to make love, pretty difficult. They need to have a way of grabbing onto each other. And certain kind of snakes have this structure, others don't. And the reason is how they mate. And this is important for mating. If you look up this in any reference book, it'll point out the fact this is not a mystery. Point out that these are critically important for mating purposes in snakes. And therefore, that's why they're there. And again, that may not be commonly discussed in herpetology books or books about snake, but it's there. I found a number of articles which did a very good job showing that these are not vestigial legs, they're copulatory organs. Same thing whales have. Whales have copulatory organs as well for the same function. So again, that's a matter of wrong information. Which is too bad. It's not. And, I just did a review of the literature and I found the articles and it said very clear. This is what they do. And so it's not wasn't discovered by a creationist, discovered by an evolutionist.
Yeah, that's another thing I appreciate about your research. I try to do the same thing. You refute their points, you refute their arguments directly from the secular literature, the conventional literature. And you can do that right from their own papers, because they'll oftentimes give you the data that refutes their position. But then in their discussion section of these papers, they'll try and provide some rescue devices, right? Some hypotheses. But that's where we can point out, no, these don't work. These hypotheses don't work. There's no real evidence for it. These are just stories. And so I really love the fact that you do that, Jerry. You're very diligent in your research. So
most of my reading I do is in evolutionary sources. Most, I would say 80% of it is in, because that's what I use to write my articles.
Right.
So therefore I do a lot of reading in there. But if you read in detail and think about what they're saying, you often realize that they're doing more to refute their own worldview than support it.
Would you say as creationists, we can get some of our best arguments right from their own literature, what they're saying?
Oh, clearly. Yeah, I do.
Yeah.
Yeah, I do.
So if you see a militant evolutionist online or in a video or in a debate, they're not as open about their errors and flaws and what they don't know, right? Because they're being an apologist for evolution. But when you go into their own literature, that's when their defenses are down and they admit to a lot of what they don't know.
That's true. They're just looking at the evidence and they're writing books on herpetology or whatever. So that's true. They're not an apologist. They accept the evolutionary worldview and they try to interpret the information according to that worldview. But on the other hand, they're not trying to push any idea. They're, maybe their wives go to church and they don't want to, some are aware of the other side and they don't want to, but nonetheless, they were at curiosity. What are these things on snakes for? What do they do with them? Why do some snakes have them and other snakes don't? What's the purpose? Right. They got that idea and they looked in the research and did papers on it and so on. They found out a good reason. So I don't think they were not trying to disprove evolution. They were not trying to disprove anything. They were just trying to understand what do these things do. Right. And they wrote papers reviewing their findings, and therefore this is just doing research. There's a lot of scientists who just don't care about creation evolution. They just look at what's there. In fact, quite a few just trying to interpret the reality that they see. And so they don't care one way or the other if it proves one view or the other view. That's not their concern. Their concern is to do science and to understand what these structures do on snakes, why they're there.
Do you think that there's a variation of designs or variations in terms of the sizes of these copulatory or reproductive features to help with mating in snakes and whales, Jerry? Because I've noticed proponents of evolution, they'll point to fossil whales like Basilosaurus or fossil snakes like Najash, and they'll point to those vestigial hind limbs, but they'll say, oh, see, in the fossil, they're a little bigger than the extant versions. So they're saying, see, so they're slowly decreasing in size. Or is it just variation in that feature, do you think, Jerry?
Well, what intrigued me most is why do some snakes have them and other snakes don't at all?
That's
and
interesting.
The research I did, it's been several years, but the research I did, they found very good reasons why some snakes have these structures and some snakes don't. And of course, if they would lose these structures for some reason, reproduction would become very difficult. So none of them that I'm aware of, that I can remember, claim they were losing them. In fact, if anything, they were getting larger because they're more effective in the copulatory matings with other snakes.
Well, that's a good point.
Not,
if they lost them completely, and the ones that don't have them, reproduction is still possible, but it's more difficult. So it's better to have them.
Much more difficult, if not, much more difficult. And there are several articles which did a good job showing why they're there, why they're needed, and how important they are, and that this is part of the structure. We need to understand why they're there. And the main, as I mentioned, the main concern I had was why do some have them and some don't?
Yeah.
And that was explained. I don't remember the details now, but they explained that quite well, why some snakes had them and other snakes didn't. Might have been water snakes are more likely to have them because they're swimming in the water, and I don't remember exactly, but the literature's there.
Yeah. Make it in the water would make it a little more difficult, which makes sense with your whales and your extinct whales too, like Basilosaurus. And so, yeah, I mean, and that's why it's such a great time to be a biblical creationist, because there's so much for us to research. And it goes back to what you said earlier about your evolutionist lenses or glasses and your creationist lenses or glasses. We can look to these structures in whales and snakes and understand, because we see treasure, we see function, we see design and intention. See, they see accident, we see intention. And lo and behold, we find a function for it. But the evolutionists, with their evolutionist lens, they see an evolutionary leftover. They see a vestigial trait. And so it comes down to a worldview issue at that point, Jerry.
That's true.
Okay, well, this has been a great show. We have touched on a lot, which was our goal. God bless you, Jerry, and the work you're doing. I look forward to having you back on in January for a debate that I'm very excited for. A lot of people excited for that. And then hopefully we'll continue our Falsification of Common Descent program series here probably in February. So, Jerry, thank you for joining us. And
Thank you.
Okay, to the audience, thank you for joining. Stand for Truth is out. God bless you all.